Uh, okay, so let's get started. So welcome everyone to this time's Zoom seminar. It's really my great pleasure to welcome today's speaker, Dr. Tony Wyss-Coray from Stanford. I really think he's a big pioneer in the field of neuroimmune crosstalk within the aging process and neurodegenerate disease. I believe a lot of us actually already benefit or learned more or less from his work for the past decades. So a very brief introduction about Tony. Tony get his undergrad study and PhD training both in University of Bern in Switzerland. And then afterwards, he started to pursue his postdoc training in the Scripps Research Institute in San Diego in neurobiology. By the way, Tony's undergrad study and PhD study is focused on microbiology and immunology. So Tony currently is the D.H. Chen Distinguished Professor of Neurology and Neurological Sciences at Stanford University, and also Associate Director of the Paul Glean Center of the Biology of Aging, and then the Director of the Stanford Alzheimer's Disease Research Center Biomarker Core. Now, his lab study brain aging and neurodegeneration with a focus on age-related cognitive decline and Alzheimer's disease. His lab is following up the early discovery, which showed circulatory blood factors can modulate the brain structure and function and factors from young organisms and rejuvenate old brains. So Tony not only being a very successful scientist, but he's also an entrepreneur who is a co-founder of Alco, has a company developing plasma-based therapies to counter age-related diseases, including Alzheimer's. Now, current study in Tony's lab focuses on understanding how the immune system and then the organism as a whole age and then communicate with the brain. Putting humans at the center of his study, Tony integrates genetics, cell biology, and proteomics approaches and models them in the short-lived killifish and in mice. Ultimately, his lab tried to understand brain aging and disease at a individual level to develop tailored diagnostic and therapeutic tools. So with his remarkable achievement in the field, he has really received numerous awards and honors, which I think is really a big, long list. Just to give a few examples, he was giving a speaker at the Global TED Royal Institution in London with 1.7 million viewers. I think it's really something stunning. And then he's also a NIH Direct Pioneer Awards receiver. Time Magazine, the Health Care 50 Most Influential People Transforming Healthcare in 2018. And then he's also got D.H. Chen Distinguished Professorship in Stanford in 2018, and he became a director in Phil and Panic Night Initiative for Brain Resilience recently. I think it's really something stunning in his CV, and we are really thrilled that you can join us today for our Zoom seminar. I think Tony is going to talk about young blood for the old brains, and we are very much looking forward to your talk. Tony, please go ahead.
Thank you, John. Thank you all for joining me in this seminar. I'm very excited to tell you a bit of what excites us, what we're interested in. And I have sort of this general title, Young Blood for Old Brains, that I've used a lot and keep updating it with new findings and discoveries. So I'll give you a few vignettes of some of our latest research, but also a background of how we arrived where we are now and what our thinking is. Just a couple disclosures. As John said, my lab is primarily interested in understanding how the brain ages and how it becomes susceptible to cognitive dysfunction, neurodegeneration and diseases such as Alzheimer's. And these are global problems. They're not first world problems, but they're really global problems because across the globe, luckily, because of increased, improved healthcare and nutrition, people get older and older. And as a consequence, they become susceptible to these age-related diseases. And of course, you know, one of the most devastating age-related conditions is if you start losing your cognitive function and you don't remember the people around you anymore, or you know, anything that you did in your life. I just give you an example of the genetics of Alzheimer's, and this could really apply to any type of disease. We have genetic information for that confers susceptibility to disease, such as in Alzheimer's disease. There are very rare mutation that cause the dominant forms, familial forms of disease. So if you have the mutation, you will get the disease in your 20s, 30s, or 40s. And then there's lots of weaker genetic risk factors from GWAS studies that confer moderate or very low risk that increase or decrease your risk to get the disease. For Alzheimer's in particular, there is the ApoE gene that has sort of an intermediate effect. 2 copies increase your risk by about 12-fold at age 65. But really, the elephant in the room for this disease is age. The sporadic forms of the disease, which affect 99.5% or more of individuals, the most dominant risk factor is clearly age. And we need to understand age in order to tackle these diseases. And this is just illustrated here. This is now not specifically Alzheimer's disease, but just cognitive decline and dementia. Now, most dementias are of the Alzheimer's type in the older people, but in the 7th decade, you find up to 1% of people who have dementia. In the 10th decade, so people to make it up to 100, at least half of them are demented. But what's also very interesting, especially from a research perspective, is that there are individuals who live up to 100 years, so roughly a third from sort of population-based studies that have cognition intact, and they perform often at a level of a 50 or even 50-year-old or even younger person. So it is possible for the human brain to remain functional until very, very late in life, up to the end of our lifespan. But as I mentioned already, it's not just the brain that suffers from age, exposure to age, but as this graph illustrates here, as people get older, they get more and more age-related diseases. So each one of these colors here indicates a number of disease from 1, 2, 3, 4, 5, and so forth. And the study suggests that around age 65, half the people have 2 age-related conditions. And you can see as they get older, you have more and more people with multiple age-related diseases. And that prompted many in the field to question whether this approach of treating each disease, such as cardiovascular disease, arthritis, cancer, many cancers are age-related, neurodegenerative diseases, whether it makes sense to tackle each one individually, or whether one could potentially target aging, the process of aging itself, and try to understand what is the molecular basis of aging, what is the cellular basis of aging, and could that be slowed down or even reversed in what people often call rejuvenation. And in this way, basically tackle multiple age-related diseases, not just one at a time. And this is really not a pipe dream anymore, I think, at least in animal models, and particularly in mice. Multiple different interventions at very different levels of biology have shown that aged animals can be treated, if you will, with age-related interventions that slow down the aging process, extend lifespan, or reverse aspects of aging at the molecular, functional, cellular levels. And I list just a few of them here, such as metabolic interventions, caloric restriction, exercise, a number of small molecules that target metabolism, the removal of senescent cells, a very hot topic recently, transient epigenetic reprogramming of aged cells, so where you try to remove those epigenetic marks that accumulate and the DNA in cells across the body with age. And then the treatment with young blood, with something that we have been focused on, as well as spinal fluid, as we recently showed. You can exchange the cerebrospinal fluid in aged mice, and you can make their brains function better and make them younger at multiple levels. So one of the primary models that has been used to make the breakthrough discoveries initially by Tom Rando, who was here at Stanford and actually recruited me and was a very good colleague and friend. He is now at UCLA, where he has the Stem Cell Institute there. But he was interested in muscle aging, muscle stem cell aging in particular, and asked whether the stem cells age because of a cellular program that determines that they will age, a cell intrinsic mechanism, or whether it's the environment that makes the cell age. And so he used this old paradigm, which is called parabiosis, where two animals, a young and an old, or just two animals in principle are surgically connected. And that leads to the fusion of their vascular system, so that you have now exchange of blood from the young mouse to the old mouse and vice versa. Now, parabiosis doesn't have to be different ages of mice, which people call heterochronic for different age parabiosis. It can be any different combination of mice, genetic models. They can be both young and so forth. And immunologists have, of course, made big use of this model to study immune cell trafficking and similar questions. So what Tom did in his pioneering studies by Irina and Michael Conboy in 2005 is show that the stem cells in the muscle of an old mouse can be regenerated and rejuvenated so that they make new muscle tissue that is very similar to that in a young mouse. And that really was a breakthrough discovery that was then followed by many others who showed that this applies to multiple other tissues as listed here. Now, Tom also showed similar rejuvenation effects in the liver, and he had indication that there's increased proliferation of cells in the brain. And that was the beginning of a very fruitful collaboration with my lab, where Saul Vileda then showed, when he was a graduate student, that the brain benefits from this, the old brain, as I'll show in the next slide. And what Saul also showed for the first time, you actually don't necessarily need the parabiosis, the surgical paradigm, but you can collect blood and even the liquid fraction of blood, which is called plasma, and from young mice into old or vice versa, by repeated infusion, replicate many of these effects. And so this is Saul. He's now an associate professor at UCSF. And so what he showed is that you can collect plasma from young mice. So again, the liquid fraction of the blood from young mice, and infuse it over several weeks into old mice. So he gave small injections of like 5% of the total blood volume every 3 days for 3 weeks. And that resulted in increased stem cell activity in the brain, increased synaptic plasticity, which is a correlate of learning and memory. It also reduced brain inflammation, very importantly, and maybe this is even a key mechanism by which the young environment actually functions in aging. And then most importantly, of course, at the functional level, it improved memory in these aged mice. And what Saul also showed for the first time is it's not just the rejuvenating paradigm in parabiosis, or again in transfer of plasma from old mice to young. You can accelerate the aging process. And I'll talk a little bit more about this at the molecular level, what happens but that causes the opposite effect. So a young mouse that is exposed to an old systemic environment with repeated infusion of old plasma or exposure in the parabiosis model has less neurogenesis, so it makes less new neurons. It has less plasticity, reduced memory function, more inflammation, and also more vascular inflammation. And this may be that interplay that connects the brain and how it responds to a systemic environment. A number of groups have identified individual factors that might be responsible for at least some of these effects, but I think this is a very short list. So here there are a few beneficial factors, a few detrimental. There's going to be dozens or even hundreds of proteins that contribute to this, and it will be interesting to figure out which ones are the key ones. But as you'll see later on, different cells may actually respond to different proteins in a beneficial or detrimental way. And so I think the cocktail that nature has produced, the blood is still the most powerful agent, and individual factors will be complicated to reproduce the whole aspects of this composite we call plasma. Nevertheless, because this was so translatable, plasma is being infused into people, across the globe, tens of thousands of times every day, in surgical procedures, if people have blood loss in an accident and so forth, but also for therapeutic purposes. If you lack, for example, certain proteins, or you have too much of a protein, people undergo plasma exchange. So we started a company, Alka, which was recently acquired by Grifols. And what companies like Grifols do, they collect plasma from thousands of people every day, and then they make different components of plasma that are used in the clinic. And so what we and Grifols did at Alka is we started clinical trials where we treated patients with mild cognitive impairment or early stage Alzheimer's disease, and infused them with plasma fractions from young donors. These are small proof of concept, safety and dosing studies at this point, but they show promise in AD and PD, Alzheimer's disease and Parkinson's disease. And then what Grifols did, they combined this with the removal of the patient's old plasma. So they had Alzheimer's patients, and they removed the plasma first from the patient, which is called apheresis, and then they replenish it with plasma from their normal collection, where the average donor age is 35. And they actually did a phase 2b, phase 3 double-blind placebo-controlled trial in 500 patients, and they can show that this slows down disease progression, improves memory, as well as daily functioning, suggesting that what we see in the mice might potentially transfer to human patients. Now, it's still early days. A lot of other groups start to do trials and test this in controlled studies in people. And we can discuss maybe in the answer question session a bit, how this could potentially lead to a treatment. But this is very exciting, but it also leaves a lot of questions, and I touched upon some of them. But how does this all work? When we first talked about this, and I gave the first talks about this, people would sort of look at me and say, this sounds too good to be true. This is sort of Frankenstein or some, crazy experiments. And how would this potentially work? There's a blood-brain barrier that prevents blood factors from getting into the brain. And all these questions and reservations people had, rightfully so, because it seems too easy that, exchange of blood would just have these dramatic effects. So what are the factors? Where do the factors come from? Which cells in the body produce the beneficial or the detrimental factors? Which cell types does the blood actually rejuvenate or accelerate aging in? How do the factors reach the brain, in particular with respect to the brain, if there is this specialized endothelial layer that prevents a lot of proteins from just getting into the tissue like it does in other tissues in the periphery? And then do cells and organs age in synchrony, sort of more questions related to aging process itself, and how do we measure aging? And I will end with this particular question, because if any of the discoveries that we make in animal models and potentially translate to humans that intervene in the aging process, then of course we have to be able to measure your relative age, and not the chronological age, which we know from your birth certificate, but we want to know, did your body age faster or slower than the chronological age? So I'm just touching on a few of these questions. To answer the question, where do the factors come from? We took sort of a naive approach at the time with Nick Schaum when he was a graduate student in the lab. And we wanted to know, where are the proteins that we had discovered to potentially be involved in the rejuvenating effects or in the pro-aging effects, where are they being produced? And one way is to look at the gene expression profile across different organs in the body. And we had the fortune to have Steve Quake as a neighbor and a colleague. And so I discussed that with Steve and said if he could help us answer this question. And he is a pioneer in nucleic acid measurements with Fluidigm and other companies he started, and methods that he developed to really measure nucleic acids on scale. And so it just happened that he became the co-director of the Chan Zuckerberg Biohub, and he had a lot of resources. And he had also developed single-cell RNA sequencing protocols in his lab. And so he was excited about this possibility and suggested we go straight into single-cell RNA sequencing rather than bulk, what we initially wanted to do. And so that led to a big consortium where we recruited more than a dozen labs at Stanford with several, at least 100 people. And we took down the mice over 5 weeks, every day, 2 mice, and gave out the tissues to all these different labs that processed them. And then we sequenced them and generated a number of resources that hopefully are useful to the community, and some on this call, such as the initial tabula muris, which was just in young mice and described gene expression across all major tissues and cell types. And then we also characterized how the transcriptome changes in the mouse with age, both at the single cell level, but also using bulk in combination with single cell, where the bulk provides statistical power and the single cell provides the resolution. And so we got a really fruitful collaboration with lots of bioinformatics and computer science people to extract very exciting new information from these. And clearly one of the key discoveries was, and this is not entirely novel, but at scale was demonstrated the first time with these studies, is that aging starts very early. It's a non-linear process, and you can sort of monitor at what age you start to see significant changes in gene expression. And if you look at this across all major tissues, you see that sort of around 12 to 15 months of age, most tissues start to show significant changes in gene expression, with downregulation and upregulation of specific types of genes. You can look at what kind of genes are these, and you can also discover which cell types show the first age-related changes. And we'll talk a little bit more about this in the next couple of slides. But it really supported this notion that aging is an asynchronous process at the tissue and cellular level. And what I mean with that is that you can't look at an organism and say the organism shows accelerated aging or decelerated aging. You really have to ask the question, which tissue and which cell in a given organism shows this behavior, because they have their own trajectories. It's not a synchronous process. It's not like all tissues and cell types start to show aging at the same time, which again strongly argues against a genetic program that age is somehow a part of our DNA. But the most likely explanation is that it's a stochastic process that is amplified by the environmental challenges that an organism is exposed to. And that jives well with, studies across different types of organisms, in C. elegans, in flies, and some of them are listed here. So what that leaves us then is still we can look at, what are sort of tissues that show the first age-related changes, and you can again, it's not at the cell level. There's cells and certain transcripts that change already earlier, but sort of globally, you could say the thymus is probably one of the first changes that tissues that shows age-related changes. Spleen, fat tissues, gonadal fat in particular, and then other tissues start to age. And this is not really, this is an illustration more so than based on, data that you can really capture and say, okay, we have to target the fat because that's where aging is most important. Very much related to this question of aging is which cells are responding to the circulatory factors that you see in a parabiosis model. And this is work spearheaded by Robbie Palovich, who is actually a trained computer scientist and studied sort of, relationships of words and all kinds of computer scientist related questions, and became interested in biology and really took this project to another level by mining the data in creative ways. But basically what he looked at is in collaboration with multiple other people, and of course with again with Nick Schaum, who started the whole project, Andreas Keller, a computer scientist bioinformatician who has his lab in Saarbrücken in Germany, and then Tan, who was at the BioHub. So in the parabiosis model, you have these mice or pairs of young with old mice. And then you have what we call isochronic, so a pair of young mice and a pair of old mice. And so as a ground truth, if you will, you have mice that are not undergoing any surgical procedure and are just, the normal aging that I just showed you before. So this is tabula muricinis, how we called it, normal aging across all major tissues and cell types. And then you can ask, so how does this normal aging compare to a young mouse that is exposed to an old environment? And we call this accelerated aging, shown here in purple. And then you can ask which genes are reversed. So genes that change with age, which ones of those are changed in the old mouse exposed to a young environment? And we call this rejuvenation in this teal color here. So again, we look at which genes show the same trajectory as in normal aging. We call this accelerated aging, and this could go up or down. And which ones show the reverse trend? That's called rejuvenation. And it's just one way to look at the aging process, of course. And so here first we look at a ranked list of cells, and we had about, so here these are the top 18 cells that I show you out of 47 cell types that we had enough statistical power to analyze. We would like to repeat this with many more cell types and more animals, but this is what we had at the time. And so we ranked them here, and there are several thousand genes that change with age in almost any cell type, from 3 months to 18 months of age, or sometimes 24 months of age. And then we can look at this accelerated aging paradigm and ask again, which cells show the same trajectory as aging in a young mouse that is exposed to old factors from the old circulation in this paradigm. And then we rank them. And what you see is at the top, sort of the cells that are most responsive to an aged environment and mimic aging are multiple types of endothelial cells in different tissues, keratinocytes, basal cells, but then also some stem cell like stromal cells and these smooth muscle cells here, as well as hematopoietic stem cell are relatively high up. But then really with the rejuvenation paradigm, where we look which ones of these genes that change with age are reversed by the rejuvenation paradigm, hematopoietic stem cells are at the top. And this is really exciting, of course, for immunologists in particular, but it suggests that stem cells are highly sensitive to the environment they live in. Because again, the only thing we do here is we are exposing old mice to factors that circulate from these young mice. And this is over a period of 5 weeks before we then take down the mice and we do the single cell RNA sequencing. You also see hepatocytes being highly responsive, as well as some other stem cell-like stromal cells. And then further down, you would have endothelial cells, but they don't make it to the very top. Interesting also, oligodendrocytes, even though they're in the brain and sort of behind a blood-brain barrier, they seem to be responsive to this paradigm. And then you can ask a further refinement, which ones of these changes are consistent with aging and which ones are reversed. And again, we find sort of a lot of responsiveness in the stem cell population, HSCs being responsive and change in the same way or in the opposite way with these paradigms. What is the biological function of these genes? And I'll just show you 2 examples here. Accelerated aging in gonadal fat stromal cell. These are cells that are highly responsive to an aged environment. And here, it's very frequently we see genes involved in extracellular matrix. And this is a common theme in the aging field, that matrix production is altered with aging. If we look at the rejuvenation paradigm, liver cells are highly responsive. And here, very interesting, and again, consistent with literature in the aging field, mitochondria are highly responsive, mitochondrial genes, or genes that are involved in mitochondrial function and electron transport are highly responsive to a rejuvenating environment, and seem to sort of contribute to that rejuvenation effect that we have observed. One sort of intriguing way to look at that is, what are the common themes of these effects across the whole organism? Are there coordinated organism-wide cellular responses to aging and parabiosis? And I show you this graph just for normal aging. This is a graph that Robbie developed that I really like. We call it the Picasso graph. And it has a lot of interesting information. Basically, what we show here is the cells that we have studied. So these are the different cell types that we have studied in this analysis, and then the tissue that they are derived from. And we show with the size of the dot, how many genes change with age in a given cell type. So there's a lot of genes changing in these larger dots here in pericytes, for example, and a bit fewer genes in some of these other cells. And then the lines that connect them basically show the similarity in the genes that change with age. And this is also in this little inset here, we show the similarity overall of gene expression with aging between cell types. So there are some cells that show a lot of genes, identical genes that change with age. And you would assume that if you have 2 different cell types and the same genes change with age, that they may respond to a cue from the outside that induces these age-related changes, or they're closely related and there's a genetic program that is responsible for that. So we don't have a final answer to this, but it's interesting to make these observations as, for example, in endothelial cells. So endothelial cells across many different tissues have a lot of similarities in gene expression changes. And the most likely interpretation is that this is induced by circulatory factors, right? That factors that are in the blood, in the plasma, they trigger programs in endothelial cells across multiple tissues that produce these age-related changes. And stromal cells are also similarly showing related transcriptional programs. And then what you find is another type of paradigm where you have cells of different types, such as in the skin, keratinocytes, epidermal cells, and basal cells that have clearly very different cellular sort of identities, but they share, in this case, similar programs of aging. And so maybe there is more of a genetic component that makes these age that is part of the sort of the makeup of the skin. But again, it's the interpretation is still something we are struggling to come up with conclusive paradigms, but it's interesting to pursue this further and make sense of this. And then you can, of course, look at this in the context of the accelerated aging or the rejuvenation paradigm. Now, other people have started to look at that at similar resolution. For example, there is a caloric restriction study where this group looked at mice or rats that had ad libitum food or were calorically restricted, and then they looked at the single cell level in 6 different tissues, and they concluded that there is single cell transcriptomes that are responsive in very specific ways to these interventions. And it will be interesting to now start comparing different interventions and see what are common paradigms of rejuvenation or age acceleration. And I think we will see a lot of studies that start to integrate these different experimental paradigms. So just quickly, what are the factors and how do we measure aging? So the blood, if you think about it, is really this tissue that connects all the different organs in the body. It's not just transporting blood cells, immune cells, and red blood cells, but it's really an organ of cellular communication. And you can use the composition of the blood at a molecular level to study physiology, I would think. And we have used it to study aging. Just one example is this study here, where we had blood samples from over 4,000 people from the interval study, as well as in collaboration with Nir Barsala and Sophia Millman at Einstein College. We had 3,000 proteins measured with the SomaScan platform. This is a platform that uses so-called aptamers to measure relative levels of thousands of different proteins. And when we do that, we can sort of visualize the changes, the proteomic changes that happen in the blood with age. And if you look at this, it's maybe less surprising why parabiosis works, why blood exchange works, because you can see how dramatic the composition changes from young people to old people. So if you give a young person this cocktail here, you can imagine this may have detrimental effects. And similarly, you know, there may be beneficial factors in a young person that have a beneficial effect. It also illustrates beautifully that aging is asynchronous. There's a lot of different processes going on, factors going up and down at different times. And again, they reflect changes throughout the body. These proteins are derived from across all cells and tissues, as we'll see in a moment. And you can also ask, when are these changes happening? When do you see significant changes in a protein between people at a given age, younger and older, let's say, than 30? And if you do that and you add up the number of proteins at each age, you get these so-called waves of aging, how we call them. So again, what this shows, for example, here, there are roughly 400 proteins. The number scale is not here, but trust me, this is about 400 proteins that are significantly different in 35-year-old people, in people younger than 35 compared to people older than 35. And then there's less proteins that change as you age, but then another sort of little peak around age 65, and then the biggest changes around age 80, a bit above age 80. You can look at what are the proteins that are in these waves, in these peaks, which are the most dramatically changing proteins. And you see that each wave has some common proteins, but they're also individual proteins that may tell us something about the biology of aging. Now, to conclude, I want to show you what else you can do with this information, because again, these proteins contain information about the physiological state of the organism. And our motivation for these studies, which were spearheaded by Hamilton O and Jared Rutledge to graduate students in the lab, our motivation was to understand, to see whether we can understand brain aging or physiological states of the brain by looking at the composition of proteins in the blood. Now, how would that possibly work? Well, the argument would be that some of the proteins that are in the blood are derived from the brain. And if we can identify them, we can sort of read out the function of the brain. And that's, of course, to some extent trivial. If you go to the doctor, they take your blood and they measure certain proteins and they tell you, your insulin level or, your glomerular filtration rates and things like that based on proteins in the blood. But we want to take this to a much higher level by using massive proteomics and looking at thousands of proteins. And then by looking at specific proteins that are tissue specific, we can guess from which tissue they are and ask whether they report function from this tissue. So the way we do this is, again, we have thousands of proteins here. We actually have almost 5,000 proteins in plasma from many, many people, thousands of people. And for each of the proteins, we can ask, where is it being produced? Now, we use the gene expression atlas, GTEx, or now Tabula Sapiens, that Steve Quake has been producing, and ask, where is a given protein expressed? Now, most proteins are expressed in multiple tissues, but about 15% are relatively specific to a given organ or tissue. And so we find about 200 proteins out of 5,000 that we can say with relatively high likelihood that they're derived from the brain, about 120 from the liver, and so forth. And so we can now ask, do these proteins, and many of them change with age, do they tell us something about aging of the brain, physiological state of the brain? And we can build these tissue clocks, as people often call them. And this is was first sort of pioneered with epigenetic changes in immune cells that Steve Horvath has pioneered, where you read basically epigenetic marks in immune cells. And what they find is that these marks change in a stunning fashion in concordance with chronological age. And then the deviation from your actual chronological age seems to be associated with biological function, and that it's often referred to as delta age or age gap. Now, we do this with proteins. And again, we do it with proteins that we believe are tissue specific because they're only expressed in that tissue or mostly expressed in that tissue. And so we built these clocks for every organ tissue with proteins that are specific to that tissue, as you see here. And then ask, can we gain information out about the biological age of each organ in a living person, as well as the physiological state? And so in this experiment, we have a training cohort. This is together with the company SomaLogic. This is actually their normal aging cohort. And we train these clocks, if you will, on the whole body. So here we don't restrict which proteins are used in the algorithm, or we make these tissue-specific heart, kidney, brain clocks. And then we test them into independent data sets, one from near Barcelona. It's called the longevity study. It's an aging and centenarian study. And then in the Stanford Alzheimer's Disease Research Center. So this is enriched with people who have early stage Alzheimer's disease, Parkinson's disease, as well as some who are demented. But the majority are actually controls, older controls. And then again, we build these clocks for every organ, and I just show you a couple of examples. So here, these are individuals where we again build a clock for the heart. For each individual, we know their actual age, and we can calculate the heart age, the predicted heart age. So this person would have a young heart, this person would have an old heart. And when we do this, of course, we know which proteins are in this clock. There's only a few that are needed. Interestingly, the algorithm recruits two clinically well-known biomarkers, natriuretic peptide B, as well as troponin, which if you are older and you have a heart problem, that's what the clinician measures. But we also recruited a number of new proteins that are involved in heart function and heart aging, it seems. And then if we go into the ADRC, if we use the organismal clock, we actually don't see a difference between people who have a pacemaker or atrial fibrillation in how old they are biologically. So the organismal proteomic clock is not useful here. But if we use the heart-specific clock that uses only these 8 proteins, we have a highly significant difference in heart age, whether a person has atrial fibrillation or a pacemaker in both ADRC and the longevity cohort. In the longevity, the organismal clock actually also works. But clearly, if you just use the heart-specific proteins, you get even a better prediction. And most interestingly is that if you look prospectively, so here we have the plasma of people. So we have a sample from people at time 0, and they're followed over 15 years. And so we have follow-up data. And when we look at the bottom 25% heart agers, so people who are here in the lowest quartile and who are predicted to have a young heart, they basically have their heart intact, and they seem to be very mildly or not affected by heart disease typically. Whereas the top heart agers, the top quartile, they have a 15-fold increase risk to develop heart disease, as you can see here. And similar data in the brain. So here again, we use now the brain-specific, restricted a list of brain-specific proteins to predict the biological age of the brain in these people. And when we look in the Alzheimer's research cohort at those who have Alzheimer's disease, 18 out of 22 are in the top quartile of brain agers, and only a few, less than a handful, are in the lowest quartile. And again, if we look prospectively, maybe the most exciting data, blood collected here at time 0. And if we follow these people over 5 years, those who are predicted to have older brains have a 7-fold increased risk to decline cognitively and show loss of cognitive function with the standard CDR sum of boxes assay. And people who are have younger brains in our model, they tend to stay more or less stable over this period of time. So these are just early data, early findings. We are now collaborating with Washington University and Carlos Cruchaga and have a new data set of several thousand individuals to further validate these findings. But you can then build these maps for an individual where you look at the relative age of each tissue in the body. And what we see again is that the brain is older in patients with Alzheimer's disease on average. But interestingly, they also have older hearts, and some other tissues seem to show accelerated aging. And there's, of course, a very well-known connection between cardiovascular disease and development of Alzheimer's disease and dementia. And what's really exciting, what we also see is sort of signs of use of specific supplements. For example, here, these are people who take vitamin D, and they tend to have slightly younger hearts, statistically significant younger hearts. People who take glucosamine have multiple tissues, seem to show a relatively younger age compared to people who don't take that supplement. And then here, multivitamin. So what we hope to do is to really use this information, refine it further, and gain information from then actual intervention, where we can look before and after an intervention. Does the age of a person change? What is the biological pathways that are in these clocks? What does it mean biologically? And hopefully in the future, recruit people specifically to clinical interventions based on the state of their tissue. And what this might look like is that if you find a person who has an accelerated brain aging, they might be at risk for Alzheimer's disease. And so you could intervene before they have the disease. People with accelerated kidney disease may develop hypertension. People with accelerated heart aging, ischemic heart disease. So with that, I'd like to thank many collaborators and people in the lab. I highlighted those who contributed to the specific data that I showed, and then funding from many different sources, and be happy to take questions. Thank you.